In 1961, Brenner and Crick realized that three-letter non-overlapping nucleotides code for amino acids. Marshall Nirenberg and J. Heinrich Mattei deciphered the first codon in the genetic code. They used a cell-free extract of E. coli, which contained all the components necessary to translate mRNA into proteins. The cell extract was treated with DNase to destroy any E. coli DNA so there would be no template DNA for the mRNA. Homopolymers, which are polymers consisting of identical monomer units, were made of uracil bases. The uracil homopolymer was added to a test tube of cell-free extract along with one radioactively labeled amino acid and 19 unlabeled amino acids. After transition took place, the protein was filtered and the filter was checked for radioactivity. They found that the codon UUU codes for the amino acid phenylalanine. They repeated this experiment with homopolymers of guanine, alanine, and cytosine. Nirenberg and Mattei found that CCC codes for proline, AAA codes for lysine, and that GGG does not code for any amino acids. Marshall Nirenberg and Philip Letter identified the remaining codons. They synthesized short mRNA codons and tested them in a similar procedure. Nirenberg and Letter added one codon to a mixture of ribosomes and tRNAs. One of the tRNA was radioactive. The ribosome bound the mRNA to the encoded tRNA. After binding occurred, the mixture was passed through a filter. Ribosome-bound mRNA stuck to the filter and unbound tRNAs passed through. The filter was checked to see what amino acids were bound to it. In this example, CCU encoded for proline. Nirenberg and Letter repeated this experiment until they were able to identify the remaining codons.